Today we're going to dive deep into one of the most revolutionary technologies in modern genomics, droplet-based single-cell sequencing. This technology has completely transformed how we study biology at the cellular level. Let me start by explaining the problem this technology solves. For decades, scientists performed bulk sequencing, where you take millions of cells, grind them up together, and sequence the average signal. But here's the problem. Tissues are not homogeneous. Each cell can be dramatically different from its neighbors. Imagine trying to understand a symphony by only hearing the average sound of all instruments combined. You'd lose all the beautiful complexity. You might ask, why does this cellular heterogeneity matter so much? Let me give you a concrete example. In a tumor, you might have cancer stem cells, rapidly dividing cells, cells resistant to chemotherapy, and cells responding to treatment. If you only look at the average, you completely miss the resistant population that will cause relapse. In the brain, there are hundreds of distinct cell types, each with unique gene expression patterns. Bulk sequencing would mix them all together and you'd never discover new cell types. Single-cell RNA sequencing solves this by measuring gene expression in individual cells. But here's the challenge. How do you process thousands or millions of cells individually without it taking forever or costing a fortune? This is where Droplet Microfluidics comes in. The key insight is brilliantly simple. Encapsulate each cell with a barcoded bead inside a tiny droplet of water suspended in oil. Each droplet acts as an isolated reaction chamber. It's typically about 100 micrometers in diameter, roughly the width of a human hair. Inside, you have one cell and one bead. Let's do some quick calculations to understand the scale. A typical droplet is 100 micrometers in diameter. The volume of a sphere is 4/3 pi r cubed. The radius is 50 micrometers. When we calculate this, we get about 524,000 cubic micrometers, or about half a nanoliter. 0.5 nanoliters. That's incredibly tiny. For comparison, a single drop of water from an eyedropper is about 50 microliters, which means one droplet is about 100,000 times smaller. And here's where it gets impressive. How many droplets can we generate? Modern microfluidic devices can produce thousands of droplets per second. A typical system generates about 3,000 to 5,000 droplets per second. This means in just 10 minutes, you can encapsulate hundreds of thousands of cells. Now let's talk about the barcoded bead, because this is the key innovation that makes everything work. Each bead is covered with millions of oligonucleotides, which are short DNA sequences. Each oligonucleotide has 3 critical parts. First, a cell barcode that's unique to that bead. Second, a unique molecular identifier, or UMI, which is a random sequence. Third, a poly-T tail that captures messenger RNA molecules from the cell. What exactly is this cell barcode? It's a short DNA sequence, typically 10 to 16 nucleotides long, that uniquely identifies which bead, and therefore which droplet and which cell, an RNA molecule came from. If you use a 12 nucleotide barcode, you have 4 to the power of 12 possible sequences, which is over 16 million unique barcodes. That's more than enough for any experiment. The unique molecular identifier is brilliant. During sequencing, we need to amplify the RNA molecules using PCR. But PCR can amplify some molecules more than others, introducing bias. The UMI allows us to distinguish between copies made during amplification and truly distinct original RNA molecules. This dramatically improves quantification accuracy. Let me walk you through the entire workflow step by step, because understanding the process reveals why this approach is so powerful. Step 1. You start with a tissue sample and dissociate it into a single-cell suspension. This might be a tumor biopsy, a piece of brain tissue, or blood cells. Step 2. The cells and barcoded beads are flowed into a microfluidic chip along with oil and surfactant. The chip has carefully designed channels that form droplets at specific junctions. The channels meet at a T-junction or flow-focusing geometry. The aqueous phase containing cells and beads meets the oil phase, and surface tension causes droplets to pinch off at incredibly high speed. Here's an important statistical point. The encapsulation follows a Poisson distribution. you can't perfectly control which droplets get a cell and which don't. The Poisson distribution tells us the probability of K cells per droplet, where lambda is the average number of cells per droplet. If lambda equals 0.1, most droplets are empty. About 10% have one cell, and less than 1% have two cells. In practice, we use a low cell concentration to minimize doublets, where two cells end up in the same droplet. This is a trade-off. Lower lambda means fewer doublets, but more empty droplets and wasted reagents. Step 3. Once the droplets are formed, we add a lysis buffer that breaks open the cell membrane. The cell's RNA molecules spill out into the droplet. The messenger RNA molecules have poly-A tails at their 3' end. These poly-A tails hybridize to the poly-T sequences on the bead oligonucleotides. This captures the RNA. Step 4. We add reverse transcriptase enzyme, which synthesizes complementary DNA from the captured RNA templates. Crucially, this cDNA now carries the cell barcode and UMI from the bead. This is the magic moment. Every single cDNA molecule now carries a label telling us which cell it came from. This means we can break all the droplets, pool everything together, and still track which cell each molecule originated from. This is why the approach is called massively parallel. We perform thousands of separate reactions simultaneously in individual droplets, then pool everything for sequencing. It's like doing thousands of experiments at once. Step 5. We break the emulsion, destroying the droplets. All the barcoded cDNA molecules are pooled together and amplified using PCR to generate enough material for sequencing. Step 6. The amplified cDNA library is sequenced using next-generation sequencing platforms like Illumina. We sequence millions of short reads, each containing the barcode, UMI, and partial gene sequence. Now you might ask, how do we make sense of millions of sequencing reads? The computational analysis is just as important as the experimental workflow. First, we demultiplex the reads by cell barcode. All reads with the same barcode came from the same cell, so we group them together. This gives us a separate dataset for each cell. Second, we use the UMIs to collapse PCR duplicates. If multiple reads have the same barcode, same gene, and same UMI, they're copies of the same original molecule. We count them only once. The gene expression level for each gene in each cell equals the number of unique UMIs we observe for that gene. This gives us accurate quantification. Third, we create a giant matrix where rows are genes, columns are cells, and each entry is the expression level of that gene in that cell. This might be 20,000 genes by 10,000 cells. Let's talk about some real numbers to understand the economics and throughput. A modern droplet-based system like the 10x Genomics Chromium can process about 500 to 10,000 cells per run. The cost per cell is typically 50 cents to $1, depending on sequencing depth. Compare this to earlier plate-based methods that cost hundreds of dollars per cell. Commercial platforms can now routinely process more than 100,000 cells per day. Some studies have profiled over 1 million cells to create comprehensive atlases of entire organs. Let me summarize why droplet-based single-cell sequencing is so transformative. It combines 3 key advantages that were previously impossible to achieve simultaneously. First, high throughput. Microfluidic droplet generation is incredibly fast. You can process tens of thousands of cells in minutes rather than days or weeks. Second, low cost per cell. By miniaturizing the reaction volumes to nanoliters and processing in parallel, reagent costs drop dramatically. This makes large-scale studies economically feasible. Third, molecular barcoding enables pooling. This is perhaps the most elegant aspect. By tagging molecules in each droplet, we convert a massively parallel physical experiment into a single pooled sample for sequencing. The applications of this technology are revolutionizing many fields of biology and medicine. Let me highlight a few breakthrough areas. In cancer research, scientists use single-cell sequencing to track how tumors evolve and identify rare, drug-resistant cells before they cause relapse. This guides treatment strategies. In immunology, researchers map the incredible diversity of T cells and B cells, tracking how the immune system responds to infections and vaccines at single-cell resolution. In neuroscience, scientists are building comprehensive atlases of brain cell types, discovering hundreds of previously unknown neuronal subtypes, and understanding their specific functions. In developmental biology, researchers track individual cells as embryos develop, watching how stem cells make fate decisions to become different cell types. Of course, no technology is perfect. Let's discuss some current limitations and where the field is heading. Current droplet methods typically only capture the 3' end of messenger RNA transcripts. This means you can count gene expression but miss information about alternative splicing and isoforms. Single-cell RNA sequencing suffers from dropout, where lowly expressed genes might not be captured at all. This creates sparse data matrices with many zeros. Despite optimization, some doublets remain where 2 cells are captured in one droplet. These create artificial cell types in the analysis that don't really exist. The future is moving toward multimodal measurements. New methods can simultaneously measure RNA, protein levels, and chromatin accessibility in the same single cells, providing a more complete picture. Another exciting direction is spatial transcriptomics, which preserves information about where cells were located in the tissue. This combines the power of single-cell resolution with spatial context. Before we wrap up, let's briefly discuss the physics that makes droplet formation possible, because it's quite elegant. Droplet formation is driven by surface tension at the oil-water interface. When 2 immiscible fluids meet in a narrow channel, the interfacial tension causes the aqueous phase to pinch off into droplets. The droplet size is proportional to the interfacial tension gamma divided by the product of flow rate Q and viscosity eta. By tuning these parameters, we control droplet size precisely. Surfactants are added to the oil phase to stabilize the droplets and prevent them from merging. Without surfactants, droplets would coalesce, and we'd lose the single-cell compartmentalization. How does droplet-based sequencing compare to alternative single-cell approaches? Let's put this in context. Plate-based methods like SMRT-seq capture full-length transcripts and have higher sensitivity, but you can only process 96 or 384 cells per plate. The throughput is much lower. Microwell-based methods also compartmentalize cells but use arrays of tiny wells instead of droplets. These can achieve similar throughput but require more specialized equipment. Split-pool barcoding methods like scRNA-seq don't physically isolate cells, but instead use combinatorial indexing. This can achieve even higher throughput, but with some trade-offs in data quality. The fundamental insight that makes droplet-based sequencing work is that physical isolation of single cells and droplets, combined with molecular barcoding, enables massively parallel processing at scale. Each droplet acts as an independent picoliter-scale reaction chamber. The oil prevents crosstalk between droplets, ensuring that each cell's molecular profile remains distinct. The barcodes are the bridge that converts thousands of parallel reactions back into a single pooled sample that can be sequenced efficiently. This is the elegant trick that makes the whole system work. Let me summarize the key concepts we've covered about droplet-based single-cell sequencing. Individual cells and barcoded beads are encapsulated together in nanoliter scale droplets at rates of thousands per second. Within each droplet, the cell is lysed and its RNA is captured on the bead and labeled with unique cell barcodes and molecular identifiers. These molecular tags allow all droplets to be pooled together while still tracking which cell each RNA molecule came from during sequencing. This approach achieves a combination of throughput, low cost per cell, and scalability that was simply impossible with previous technologies. The technology has fundamentally transformed our ability to study cellular heterogeneity in complex tissues, leading to discoveries about cell types, states, and transitions. At its core, droplet-based single-cell sequencing showcases the power of combining microfluidic miniaturization with molecular barcoding to solve problems that seemed intractable just a decade ago.